Hi, I'm Yuanyuan. Let's talk about how to breed new cannabis varieties more efficiently. Traditional breeding has been going on for hundreds of years and is still commonly used today. Growers use traditional breeding to generate new cannabis varieties with favorable characteristics, such as high yield, drought-resistant, pest-resistant, and so on. However, there are some drawbacks and limitations in terms of traditional breeding method. The traditional breeding is based on controlled mating by selection of parent plants presenting desirable traits that can be combined in the offspring. The resulting offspring are evaluated to see if they have desirable characteristics. The identification of desirable offspring is laborious and time-consuming, while some desirable traits are affected by environmental factors. For example, the stress-resistant varieties cultivated in the northern area where it's cold and dry may not have significant stress resistance when being planted in the southern area where it's warm and humid. The continuous application of traditional breeding methods in a given species could lead to the narrowing of the gene pool from which cultivars are drawn, rendering crops vulnerable to biotic and abiotic stresses and hampering future progress. Consequently, traditional breeding strategies have made limited progress in obtaining the new hybrids or varieties. To solve these problems, we apply advanced molecular breeding technology to generate new cannabis varieties. First, we find the desirable traits-associated genes and their coding sequence. Then, specific primers are used to amplify these genes in vitro. Subsequently, these genes are introduced to a linearized vector to generate recombinant plasmids, which are transformed into competent E. coli cells to amplify. The resulted clones are selected for sequencing to verify the successful cloning. Afterwards, the plasmid containing the traits-associated genes are extracted and transformed into agrobacteria, which then infect the cannabis callus. Finally, we can get the plants with desirable traits through tissue culture. This is our new high CBD cultivar. The CBD content is higher than 12%. Taken together, molecular breeding pinpoints the genes related to desirable traits and monitors the selection of specific genes or marker profiles. As a result, we can create new cannabis varieties more accurate and efficient and fit different needs in a rapid breeding cycle.